right, it's been 2 minutes, so I'm going to get started. Welcome and thank you very much for joining us for the 4th webinar of the National Early Detection and Rapid Response Framework webinar series. I'm Kaylin Clements. I'm a social scientist for the U.S. Geological Survey. I'm also the Partner Engagement Coordinator and one of the co-chairs for the National eDRR Framework, which is an initiative to strengthen interjurisdictional coordination and capacity to efficiently detect and respond to new invasive species across the nation. This series is a collaboration between the Association of Fish and Wildlife Agencies and the Department of Interior to share information about the National eDRR Framework and discuss important topics with state agencies and other organizations involved in eDRR and invasive species work. Carrie Wickstead, who's typically with me on these webinars, the former invasive species program manager for AFWA, though she's not co-facilitating with me today, she did work closely with us on the series and planning this webinar, and we're very thankful to her and AFWA for their collaboration on these. And I want to introduce Mark Frey, my co-facilitator today. Mark is the coordinator for the National eDRR Framework and will be sharing responsibilities with me, like monitoring the chat, sharing some links and materials, helping troubleshoot, etc. So I just wanted to point that out in case you see messages from Mark coming through or if he needs to chime in. In addition, we have more than a dozen volunteers from across DOI who are helping facilitate today's breakout rooms, and big thanks to them. And thank you all for being here. We've received a lot of interest in today's webinar from across geographies and across sectors, and it would be great to learn a little bit about where you all are coming from. So if you could take a moment now to post in the chat, and oh my gosh, we have over 100 people, so this will be a huge chat. Please put your, and I'll post this one after I say it, your name, your title or job, what organization you're affiliated with, and any experience or interests related to eDNA if you like. If you happen to be tied to the eDRR framework, throw that in there too. I'm going to post that in chat. I know it's kind of hard to remember. And by the way, if you can't see the chat, click the chat icon on the bottom of the screen, which I'm working on doing for myself right now. And then it should pop up. To give you some context around today's webinar, like I said, it's the 4th in the series. The first 2 webinars were last spring. The first included a foundation for understanding eDNA and its application for eDRR, as well as small group discussions on environmental DNA with the audience. If you missed it and you'd like to watch it, please see a link, which I'm going to post right now. It's called a Wakelet. It's pretty cool. Here we go. And I'll tell you more about it after I paste it because I cannot multitask. So anyway, so this Wakelet will have a bunch of materials as well as recordings of old webinars at the bottom, and then eventually we'll post this recording at the bottom as well. Like I was saying, we had that first webinar. Then the second one focused on horizon scans and hotspot analysis and included discussions with the audience about how to make a public-facing online hotspot analysis tool useful for on-the-ground applications. And that tool is under development, will be posted on hosted on Siren. And the third webinar was last December and focused on molecular resources provided through the National eDRR Framework, such as the Molecular Lab Network, as well as examples of applications of eDNA for invasive species surveillance in the Great Lakes. And I just dropped a line about Siren without explaining what it was. I don't have a ton of time to. Mark, could you put a link to Siren in the chat for me? Thanks. Today's webinar will begin with a presentation on eDNA point of use tools, including applications in Voyageurs National Park. Then we'll learn about eDNA automated sampling with presentations on applications in Colorado and California. Thank you, presenters. Then we'll go to breakout sessions to hear from your perspectives on potential applications for these eDNA tools. Our agenda and presentations today actually address many of the topics and questions you submitted when you registered, so thanks a lot for that feedback. And after our point of use presenters, we'll have Q&A, and then after the automated sampling presentations, we'll also have Q&A. But please feel free to post any questions or comments you think of while the presentations are going on. Don't hesitate to put them in the chat. Mark and I will keep track, and we will use them during the Q&A for discussion. And then we'll disperse into breakout rooms for facilitated discussions on questions that we kind of pre-prepared. Um, Please keep yourself muted during the presentations to avoid background noise. Everyone's muted right now, but just keep that in mind. And with that, I'd like to move on to our first presenters. Our first presenters are Steve and Ryan Mackey. Steve is a research biologist with the USGS Upper Midwest Environmental Sciences Center in La Crosse, Wisconsin. His work focuses on developing and testing genetic tools for species monitoring and management, with a particular focus on eDNA methods for invasive species, such as the Point of Use project under the EDRR framework. And Ryan Mackey, since 2004, has served as an aquatic ecologist at Voyageurs National Park in Minnesota. He routinely collaborates on research focused on harmful algal blooms, mercury contamination, fish, the impacts of aquatic invasive species, and the effects of water level management on the park's aquatic ecosystem. And with that, I'm going to share my screen with everyone's slides are all compiled together on my master slide deck. So I'll be the one sharing my screen real quick. Bear with me. How are we looking, Steve?
Looks good.
We'll take it away.
All right, thanks, Kaylin, and thanks everyone for showing up. So yeah, I'm going to start off by talking about what point of use eDNA is, and then kind of give an overview, including some highlights of some of the research and development that we've done to bring these tools to be able to be used in the field. And then, as Kaylin said, Ryan will talk about a specific example of their experience using this protocol at Voyageurs. Next slide. So point of use eDNA, at least the way that we're defining it, is pretty broadly. It's just any ability to collect and fully analyze an eDNA genetic sample on site, presumably for things like rapid surveillance, because you would expect that if you want to do it on site, that the timing is going to be really important, that you need an answer right away. And on site can vary from, a remote lake to, your office or whatever, or an airport, whatever makes the most sense. It's analogous to medical point of care. So, everyone remembers going to the COVID test sites, and you could maybe get the PCR test that, you'd have to wait a little bit to the result, but you also could get the rapid test. And so this is analogous to that. And for these things to work, not only do they need to be able to be done at site, hopefully quickly, but also require minimal training, because you're not expecting like a band of geneticists going around to do these. Managers and other folks need to be able to run these. And you can think about a lot of potential applications for invasive species. So certainly surveillance at different points of entry, trying to catch invasive species that might be hitchhiking or such. Monitoring different events where management is time sensitive, if you need to detect it and then be able to do a management action right away. And then just, it can be useful certainly for people that don't have an easy access to labs or just working in remote environments. Next slide. So I'm first just going to very quickly go through what a typical lab-based eDNA workflow. And obviously this is going to vary depending on labs and projects, but this is I think a general characteristic. So, first there's rapid field collection for the source, typically in the field. But then those samples are sent to a lab, however they're collected, filtered, or otherwise. And typically the DNA extraction step that's used, takes a few hours in the lab. Usually the most common one, for instance, you would run it through a filter, through multiple steps, different buffers to just isolate the DNA with centrifuges and that sort of equipment. And then the gold standard for running eDNA reactions currently is quantitative PCR. And so those would be set up making a reagent mix stored in the freezer just before you set it up in the lab and run on plates. And then the equipment is going to be a thermocycler. So it can do variable temperatures, because PCR requires cycling amongst different temperatures, with a fluorometer. And all that means is the ability to detect a fluorescent dye that's in the reaction to see if you have a positive or not. So just very briefly, that's a typical eDNA workflow. You don't need to understand everything that I just said, but the key thing is that there's certain components that are really difficult to do outside of a lab environment with the typical workflow. Oh, sorry, next slide. So to adapt something like that to a point of use, Some of it's going to be the same, but there's some things that we need to change for it to really be effective. So the field collection, that probably isn't going to change at all. So that first component is likely to be the same. But for the second component, you need some sort of portable DNA extraction, that doesn't require large, bulky centrifuges or other equipment like that. And ideally something that's not going to take multiple steps and an hour or two to do, but something that you can do in just a few minutes. For the DNA test, so where the spot where I was talking about the PCR on the previous slide, so you don't want to have reagents that you have to keep in the freezer just before you run the reaction. You want to be able to have field stable or ambient stable DNA tests. So something that even if long-term storage is in a freezer, that you can have out with you for the day and it's still going to work. And then you need some sort of portable amplification device so that you would run on this, that you would actually run the reaction, but that you could easily carry with you. So next slide. So this is what we've been working on, taking a typical eDNA protocol, lab-based protocol, in adapting it to work in the field. So the first big component is that rapid extraction. And again, our goal here is really, although you could do point of use and have it take some time, our goal is really trying to make this as fast as possible, less than an hour and even faster if we can. And so there do exist some different rapid DNA extraction protocols. And we tested a couple of two general types. So the first is just using an extraction solution that releases the DNA through heating it. And depending on the exact solution, the mechanism might be different. There's two in particular that we focused on. One is a resin called Helix that basically binds non-DNA components. And so you just have DNA in the reaction that doesn't bind to the resin. And then there are commercial extraction solutions that are breaking apart the cells, releasing the DNA, and breaking down the parts, cellular components that aren't DNA. The second broad type is a syringe-based method. And this is really analogous to what we're doing in the lab with running the DNA sample through a filter and capturing the DNA that way, except it's just miniaturized and the filter's in a small syringe. So it typically does have multiple steps because you're running it through a few different buffers, but those steps are pretty quick, and you can do these within a few minutes. So we wanted to compare these two because they're all portable. In the case of the DNA released through heating, you would need a heat block, but there are portable heat blocks available. Next slide. So I understand this is a little bit of a busy graph, but there's really one thing I'm going to focus on for the purposes of today. And so we did an experiment where we spiked known numbers of cells on filters and then ran them through these different types of extraction methods. So both a couple of the syringe type methods, a couple of the heated solution methods, and then actually our lab centrifuge method. And what we found, and the thing to focus on, is if you look on, we did this by samples that were collected through both a mesocosm in our center and also from a river. But in each case, if you look on the right, those two bars by the heated solution always had, on average, the highest number of yield from the extraction, the highest number of copies per reaction that we ran. As you can see, there was a fair amount of variation compared to the other methods, but they were always higher. And interesting enough, the Helix method, the CH that's on the far right with the river, was actually less variable and consistently higher than everything else. So this tells us that these very simple heated extraction solution method, so where you're just taking your filter or whatever how you're collecting it, putting it in this liquid, heat it up for 5 to 15 minutes, depending on the method, and then that's giving you potentially a lot of DNA that could work for the next step, the genetic test. So that was really exciting that the simplest method seemed to be the best. No, that's great, Kaylin. Next slide. So then, of course, is what type of genetic test? So I talked about qPCR being the gold standard. However, qPCR requires equipment that changes temperature, that cycles temperature precisely, and also it takes at least an hour or so, depending on the exact protocol. LAMP, which stands for loop-mediated isothermal amplification, is a genetic test that simplifies some of this. First, as isothermal in the name indicates, it just needs a single temperature to run, so that simplifies the type of equipment. At its basic, you could potentially do it with a heat block as long as you had some way to assess whether the sample was positive or not. And it's also very rapid. typical LAMP reaction will, if it's going to work, it's going to amplify within 10 to 15 minutes. So, quarter more or less of the time than PCR. And it's been used a lot with the medical point of care. So Ebola, COVID, other diseases, these have been developed for, and that it's worked well. The trade-off is that it does tend to have lower sensitivity than qPCR in the sense that qPCR will typically, for a given species, detect a lower copy number. So that's something that needs to be tested to see if these are still useful in the field if they have sufficient sensitivity. Next slide. And of course, I talked about how the reactions need to be stable. So even with LAMP, if you're running it with standard reagents, they still need to be kept in the freezer, kept cold before you start the reaction. However, we tested, could we then lyophilize or basically freeze dry these reactions so that they would be field stable, and would they still work? And what this graph is showing is that yes, the lines that just basically go up and then plateau, that's showing a successful reaction, that DNA was there of our target. On the left, this is for an assay we've developed for zebra mussel. And you can see on the bottom what the lyophilized reactions look like. So on the left is the control. This is the reaction solution that has to be kept cold. And on the right is after it's lyophilized. And you can see they both work, and they both work in less than 10 minutes. So we have a field stable way of running these. Next slide. So we're taking this general protocol and we're working with a number of different partners to test these out for different targets that we're and our overall project team are developing in our labs. So some of the ongoing ones, we've been working with multiple partners out west to trial these for testing for dracinid mussels using two separate zebra lamp assay and a quagga lamp assay in different western lakes using filtering as an early detection tool there. We've worked a lot with different partners, the National Park Service and University of Montana and the upper Midwest and different rivers and lakes. In this case, using plankton toe samples. So in the top right side, you can see what a toe sampler looks like. These are often used by folks looking for zebra and quagga mussel veligers, for instance, in adapting our protocol for that. And I can talk more about that in the Q&A if you're interested in that adaptation for that protocol. Also with other targets such as snapping turtle in the lower Colorado and Arizona, and with UW Stevens Point, red swamp crayfish in Michigan. And we have some upcoming work to adapt these to airborne eDNA for terrestrial targets, particularly in the Pacific Islands, as well as eRNA, where we would be expecting to just get live organisms for testing the success of eradications in different sample types. Next slide. Oops, sorry. So I just want to close out my part for just some general considerations for if point of use might be good for your applications. The biggest thing is really if time to result, time to getting that test run is critical. If you need to have a rapid answer. If you don't need a rapid answer, if you can wait a while, days, weeks, then it's not likely to be as valuable. But if you need to know right away or really fast, this is what these are designed for. They are low throughput. And by that, I mean, you're only running, one or two samples at a time for, at most like one or two species targets. So you're not going to be able to run through a lot of samples if you're doing large scale monitoring. This isn't really eDNA per se, but I think it's especially useful if you are doing, say, testing, for trying to figure out what species is of like, some fish or mussel that you find, at an inspection or something like that, because the sensitivity then is not going to be as much of an issue, and you'll be able to identify what the species is. Right now, we've had really good success with less complex environments. We're doing some testing about adapting the protocol for more complex or turbid environments, but again, I can talk about later if folks have questions. And I think at this stage, it's really helpful if you incorporate other methods to complement. Ideally, pairing these with qPCR samples where they can be run later in the lab and help to assess how well these point of use assays are doing for sensitivity. And with that, I'll turn it over to Ryan.
Thanks, Steve and Kaylin. So I just have some quick notes on field implementation of this method at Voyageurs National Park from last summer. For those of you not familiar with the park, we're located on the US-Canada border up in northern Minnesota. You can go ahead and go to the next slide, Kaylin. So I had a couple of kind of recurring themes throughout this. One was that we had a really full field season this year. We had a lot of funded research on top of our routine monitoring, and so that kind of was a big load for our seasonal technicians and interns as far as learning and mastering methods. And so that meant that the in-person training and detailed protocol were really invaluable and necessary for this to be successful. Um, and I also really like to mention that an additional benefit was having Steve come to the park and work directly with our seasonal staff and interns. We try to do that with as many of our projects as possible. Just a great experience for them, sort of a mentoring situation, and also just for them to learn a lot of technical detail about these. They really appreciate it. So that was really cool too. Next slide. So I wanted to point out that some of our field sites were very easy logistically to sample, and others were really difficult. So here's one of the easy ones. You can see they're in a large motorboat on a lake. No real challenges other than taking a bunch of gear out there. Um, we also sampled a lot of marinas, which are really likely infestation points or introduction points, of course. So a good place to monitor. And again, that's very simple. We're generally standing on docks and taking samples, and it's easy to accomplish your sampling. Next slide. However, being like most national parks, we have a lot of remote areas as well, and we like to sample. Some of those are heavily used, believe it or not, and we like to sample for invasive species in those locations as well. So those can be much more logistically challenging. Next slide. So keeping on that topic of logistics and planning, again, with our really booked summer, I think our seasonal technicians had to accomplish about 20 different protocols during the summer. Um, and so that's a lot for these, generally these are new employees each summer. So that's a lot for them to take in. We maintain detailed protocols, both electronically and on write in the rain paper so they can take it out to these different field locations with them. But again, being that busy and having to do something different every week and maybe 2 or 3 different things during a week, the detailed protocol and the in-person training were critical to this being successful. And then I really felt like the good news was the sampling and lab work for this method were easily accomplishable for our seasonal technicians and interns once they were trained. So as far as a field test, I think this was really successful, and it's not beyond their capacity in any way. So that was excellent. Next slide. So I was asked to talk about what gaps this might fill in our monitoring and what the potential applications of this method are in our park. So some of these are pretty obvious, but they could alert us to aquatic invasive species in a marina earlier than some of our other methods. We would probably want to still verify physical presence of the species if we did that or had that sort of eDNA hit. But that could potentially allow us to start a rapid response right away too, such as closing off a marina and treating for zebra mussels. This is a great method for areas at high risk of introduction and other areas, and in areas where other methods might be challenging. And we do a lot of existing monitoring for a variety of invasive species. We have several in park lakes already. But this does add a novel method. It adds another, it complements what we're doing. And I think the last point here is if it ends up being the case that this point of use eDNA sampling is more sensitive than our other methods, then of course it would have great value in improving our opportunities for making a rapid response to an infestation. Next slide. So I was also asked to cover what happens if we detect, like what's the park going to do if we detect invasive mussels? So again, if we detect them with this eDNA methodology, we want to confirm the invasion by physical methods. So we've had a scuba team in the park before. We could very quickly get out there with snorkel gear and try to get down there and sample, etc., and take veliger tows too, and get those looked at very quickly by our partners. So we would try to attempt to confirm the invasion by physical means, like I said. We would also communicate this really quickly with our partners. Again, noting that we're on our large lakes are shared with Canada. We have almost a crazy number of collaborating agencies with jurisdictions that overlap on these lakes. And we're in regular contact with them for a whole variety of different reasons. So it would be very quick and easy for us to communicate this result with all of those people and entities. And also we have draft press releases ready to go to let the public know as well, so that we don't have to lose time developing those. And that's sort of part of our rapid response planning process, which I'll get to in a second as well. And then depending on the location of the infestation or detection, we might attempt a rapid response as well. So if we're able to have a good idea that it's confined to a specific area and that's near shore or in a marina, for instance, we might be able to deploy a rapid response, which would include, tarping off, if it's a marina, tarping the marina off and treating. We do have some of those supplies on hand already. We had a rapid response plan that was nearly complete. And we had, purchased these supplies and gone a long way towards starting to work on compliance and everything for it. But we did have a detection of veligers in one lake out in the large part of the lake, and that kind of threw a curveball at our plans. So we're in the process of revising that, but I do anticipate we'll complete a rapid response plan soon again and be ready to take action if we do have a detection. And then one more slide. You can go ahead. Yeah. All right. So then the last slide is just lessons learned from our field testing of this last summer. Thankfully, I'd say nothing major. We didn't have any major missteps. I think, again, due to the heavy workload of the seasonal employees and how many different tasks or methods they were carrying out all the time, we may have provided more examples of minor errors than we would have liked to. One employee got a little overexcited and lost part of the hand crank centrifuge to the bottom of the lake. We also had to fill in some details on the field and lab protocols early in the season, which is typical. Again, they all need to kind of be spelled out when people are doing so many different things. We did smear the labeling on some of the micro centrifuge tubes, and we lost, ability to identify which samples they were on a few of those. And our crew came up with a backup system to kind of prevent that. And then Steve let me know as well that going into this field season, they actually have barcode labels for the microcentrifuge tubes from the LIMS system. So this one's really a moot point moving forward. And then the last thing was we had a little bit of a bottleneck in lab processing due to capacity of the portable amplification device we had. So having either more of those units or the newer higher capacity version would have kind of helped things go more quickly in the lab. But as I said, I think overall it was very successful, and that's the end of what I have. So I'll take questions when we get to that point. Thanks.
Thanks so much, Steve and Ryan. I don't know why my camera is not coming on, but it looks like we have a few questions in the chat for you, and that's what we'll do now. We'll take some time for those. It looks like, Steve, you and others are already answering some of these questions, but I wonder if we could, in case people aren't monitoring the chat, kind of summarize what you all have been chatting about. Looks like the first question was an extraction kit used in the lab. If so, what kind? And Steve, would you mind kind of summarizing what's going on here in the chat?
Yeah, and there were response by my fellow lab mates on this, but yeah, the lab method that we compared it to, the specific one is it's the IBI centrifuge kit, but it essentially works the same as the Kaijin one that a lot of people are familiar with. And what it is, it's the small filter that you would then add the sample to, centrifuge through. The filter would contain the DNA, and then you have some different wash buffers that would, at the end, you would have purified DNA. So it's a pretty typical laboratory style extraction method. The multi-step takes more time that we were comparing to the rapid methods.
Thanks, Steve. There's a question that I can easily answer. Is there going to be downloadable slides? We don't have slides, but we'll post the recording, and you'll see the slides on there. The next question was, Oh, they're coming in, so it's hard to do. Okay. Is this similar to the Lucira COVID flu test that heat the vial sample or the vial solution?
And it looks like Chris Merkus looked it up, and yeah, it looks like it is a LAMP assay, just the RNA LAMP because of COVID being an RNA virus. But yes, it's basically the same sort of protocol or same sort of test.
Thanks. And thanks for repeating this from the chat. I don't think they get recorded. So it's really helpful for longevity. So then the other question was, is there a downside of waiting for results rather than using the LAMP technique for immediate results, for example, sample preservation, degradation, and inhibition?
And no, not necessarily. I mean, certainly there's a lot of good methods for preserving eDNA samples after collection that you don't need to run them right away, like that they're perfectly good to send to a lab and that DNA is going to be preserved. Some may be better than others, but yeah, there are well-established methods for doing that.
Thanks, Steve. And there are a few more similar to those questions, but I'm going to give you a break. Ryan, there's a question specifically for you. Were there any lessons learned depending on water turbidity, plants, organic matter with clogging filters, or was that never an issue?
We did have definitely some sites where we had a lot of algal bloom activity or harmful algal bloom activity later in the season, and that kind of added a lot of junk to the plankton nets, but I'm not sure if it really had any impact on the actual processing. Do you remember that, Steve, whether it actually impacted the processing or not?
I mean, there were some that, yeah, like at least when I was out with y'all and with other tow nets where there's a lot of material. And so it was unclear, so the samples were dirtier, for lack of a better term, from all the material in there. One of the things I'll mention real quick that I wasn't able to get into just because of time, but that we have discovered the tow samples is that in some tow samples, we have detected inhibitors. So things that are blocking the LAMP reaction from working, and we have ways of determining if that's going on. We found, however, if we've diluted those samples, that generally they then work. So one of the things we're working on the lab testing out right now is if we add more extraction solution from the get-go for those sort of samples, if that does the dilution, but also the extraction, so you don't have to do a second step. So that is one thing that we're still figuring out with the tow samples. If you have really highly concentrated ones.
Thanks, Steve. And I don't think we have time to go over all of the questions that came up in the chat, but I just want to note a comment that I think everybody's kind of aware of, eDNA. Monica, thank you for this comment in the chat. She said, eDNA is not necessarily indicative of a live organism. I'd have difficulty with actually initiating a treatment without confirming invasion. We respond to eDNA by increased sampling or taking it as a caution flag if sampling is already high. A few people agreed. And Chris from Steve's lab mentioned, I think this is a prudent approach. It comes down to your risk tolerance. It may take, I'm sorry, this chat keeps moving as people add things. I think in many situations, an invader may come in too small of numbers to establish or signal could be from nearby. If you have more questions, please feel free to put them in the chat. And it looks like Steve and his team are very much on top of that. But for the sake of time, we'll move on to our next presenters. And I'll get my presentation, not my presentation, their presentation ready. One moment. So our next presenters are Adam Sepulveda and Robert Walters. Adam is a research zoologist at the U.S. Geological Survey, Northern Rocky Mountain Science Center in Bozeman, Montana. His research focuses on the development and validation of tools to support biological threat surveillance. And Robert Walters has been with Colorado Parks and Wildlife's Aquatic Nuisance Species Program since 2012 and has served in the role of Invasive Species Program Manager since 2020. Thank you both for being with us. And give me about 5 seconds and then share slides. How are we looking, Adam?
Thank you. Yep, looking great. Thank you, Kaylin.
Thanks.
So in In my brief presentation today, I'll provide an overview of eDNA autonomous or auto samplers before transitioning to Robert and then another set of partners from California that are also using eDNA auto samplers to inform their own biological surveillance programs. Next slide. So very common problem for many of us. Natural resource managers are challenged with monitoring rare taxa, such as emerging invasive species and even imperiled species, so anything that's rare, across very vast spatial and temporal scales, as shown here with this map from showing all the different Park Service units across the country, or even within a state in California, showing how many state parks they have. And so we can consider specifically the case of Dreissena mussels, where monitoring often occurs near public access points when water temperatures are between 12 and 30 degrees Celsius, which is when they are potentially reproductively active. So when we think about monitoring, this could mean monitoring at tens of boat ramps and marinas at a single water body for multiple months each year. And then for oftentimes states have hundreds of water bodies. So it's just a huge challenge to manage across these vast spatial scales, but also temporal scales to ensure we have high probabilities of detecting something when it's at low abundance. Next slide. And this challenge is this too much because most agencies are limited by human resources, not to mention funding. There aren't enough of us to do the monitoring at as many times and places as is needed to be consistently effective. Moreover, conventional monitoring approaches often take multiple people, especially when we're looking for a needle in a haystack. For example, a small New Zealand mud snail. Next slide. If we are less effective or under effective in monitoring, this is a big problem because the longer an infestation goes undetected and untreated, the more likely it is to cause adverse impacts like limiting recreational opportunities like boating and fishing, degrading ecosystem health, and even harming human health, such as the harmful algal blooms that was mentioned in the previous talk. Next slide. Environmental DNA, eDNA, and the integration of eDNA sampling with autonomous sampling platforms provides a potential solution to this problem. Environmental DNA methods can be a very sensitive and scalable approach for monitoring hard-to-find organisms. However, collecting water samples by hand requires personnel or human resources to be at the right time and the right place. But we can overcome these human resource constraints by integrating eDNA sampling into auto samplers. At this current time, auto samplers collect, filter, and preserve an eDNA water sample. They do not do in situ analysis or real-time analysis of that water sample. Samples still must be transported to the lab for analysis. Auto samplers can be just deployed streamside or even within a water body and can be programmed to autonomously collect and preserve water samples at any desired frequency, hourly, daily, weekly, whatever is needed. A technician then visits the auto sampler at infrequent intervals to collect the samples and ship them to a lab where they can be analyzed. Next slide. Luckily, there are several eDNA auto samplers that are now commercially available and many others that are in the development and validation pipeline. It is best to consider these auto samplers as a set of screwdrivers, where each one has an optimal fit for purpose, just like a Phillips head screwdriver works best for a specific type of screw. When the tool and application are appropriately matched, we can cook and find these invasive species. Next slide. eDNA auto samplers have many benefits. They can collect tens to hundreds of samples autonomously in between technician visits, as demonstrated in this figure where we deployed an auto sampler on the Yellowstone River on approximately July 15th. It then collected 132 samples at sub-daily and daily frequencies, and we then returned to pick up those samples in October. Sampling is not constrained by safety, weather, work schedules, or the number of people that can get to the job site. And moreover, most auto samplers are now being designed to be easy to use with minimal training. And they also provide a way to standardize field sampling protocols across survey events that occur across different crews, so across space and across time. Next slide. Validation efforts of auto samplers are critical to determine the fit for purpose and have demonstrated that auto samplers can collect high quality samples that are equal to and at times better than manual methods of eDNA sampling and filtration, such as those collected by a Smith Root backpack sampler or a streamside peristaltic pump or even a hand pump. And this is demonstrated in this figure here where we compared these different methods. Next slide. The USGS has begun to get these auto samplers into the hands of natural resource managers in multiple national parks and 2 states where they seldom have the personnel needed for high frequency eDNA sampling or even conventional non-molecular sampling. USGS is providing the training, study design support, and lab analyses, but the managers are doing on-the-ground work. So far, the responses from managers have been very positive and indicate that auto samplers provide enough added value to be worth it. But don't take it from me. Let's hear from Robert and his experience. So we can now transition on to Robert. Thank you.
Yeah, thank you, Adam, and thanks, Kaylin. My name's again Robert Walters, and I'm the Invasive Species Program Manager here for Colorado Parks and Wildlife, and I will share with you our experience utilizing one of these autonomous samplers on the Colorado River following a detection of zebra mussel veligers. Next slide, please. Before I get into the use of the autonomous sampler itself, just to provide a little bit of background information regarding how we got here. So in July of this past year, we detected zebra mussel veligers in both the Government Highline Canal and the Colorado River near the city of Grand Junction on the west slope of Colorado. We had 3 distinct findings of mussel veligers in the Government Highline Canal and 4 in the Colorado River. Based upon these findings at different locations on different sampling dates, we considered both the Government Highline Canal and the Colorado River positive for the presence of zebra mussel veligers. Next slide. The finding of these veligers really triggered our rapid response plan, a primary component of which is doing everything we can to determine both the source and the extent of these mussel veligers in the Colorado River system. We put forward a significant amount of effort to try to answer those exact questions. Some of what that looked like is we had a raft-based delimiting survey that stretched all the way from Glenwood Springs out to the Colorado border. We did that over the course of 2 weeks in coordination with the U.S. Fish and Wildlife Service, as well as internally within CPW. In that sampling effort, we collected 95 plankton tow samples, which we processed in-house and in coordination with the Bureau of Reclamation's Technical Service Center here in Colorado. Following that initial delimiting survey, we continued to do weekly plankton tow sampling on the Colorado River from DeBeck all the way down to Grand Junction. And in September, we did a second delimiting survey, again, really stretching from Rifle down to Grand Junction. In total, we collected more than 400 plankton tow samples in the Grand Junction area, the vast majority of which were split and evaluated both via microscopy and in-house qPCR. Despite all of that effort, we found no additional evidence of mussels anywhere in the system, including any evidence of eDNA, veligers, or adults. Next slide. This is a little bit of a challenging situation to be in as an invasive species program manager. Of course, we want to know where these are coming from and how far they have extended so we can consider possible actions. Based upon the fact that we were not finding anything with these more traditional or conventional plankton tow methods, I had heard Adam present many times about these autonomous samplers in various places. So I reached out to USGS to see if they thought this might be a good fit for what we were doing out there on the Colorado River. We came to the conclusion that this might in fact be a good supplement to what we were already doing, with one of the primary components being the fact that we could increase our sampling frequency from once per week, which is what we were able to achieve with our technicians, up to once per day. We were very fortunate that there was one of these autonomous samplers already deployed pretty close to us here in Colorado out at Dinosaur National Monument. USGS, the National Park Service, were both very great to work with, and we were able to get that equipment moved over to the Grand Junction area where we deployed it on the Colorado River below the Cameo Roller Dam on September the 5th. I would like to highlight, you can see the blue spot there on the map. This is a very secure area where we were able to deploy this particular piece of equipment. This is on a piece of property that's managed by the Grand Valley Water Users Association. We thought this was really important as this equipment was on loan to us and is relatively expensive, and we wanted to make sure it was in a very secure location. Next slide. So, what did we find? How did this work for us? So from September the 5th until the middle of October, the autonomous sampler collected 46 filters. While we were working on this, we also continued along with those more traditional sampling techniques all along the Colorado River system. What we ultimately ended up finding is that one of these filters did come up with a positive result for zebra mussel DNA in one sample that was collected on September the 14th. The plankton toes that were collected adjacent to the autonomous sampler, not on the same day, but once per week, did not provide any positive results. Next. So having used this equipment in this particular application, at this point, what do we see as some of the benefits and challenges of this autonomous sampling technology? First, I would say that it was very simple to install and operate out there on the ground. All of the work that was performed, whether that was installation or the actually tending of the technology, was performed by field level technicians with very limited to no exposure to this sort of technology or eDNA work in general. We were able to get this thing installed after only a couple-hour consultation with Adam and USGS as well as Smith Root. And in terms of actually tending the equipment, we followed a written protocol, and it was that simple. This did effectively increase our sample quantity as we had hoped. We were able to get up from that one sample per week up to one sample per day. Since we already had a technician going out there on a weekly basis, there was very limited addition to his existing workload to essentially swap out those filters and collect the necessary data. Once again, we did have this positive result when the plankton toes did not produce them, so certainly something to consider. Of course, nothing is perfect. We did have some challenges in utilizing the equipment. First, as I mentioned, we were really limited on the deployment locations, really just in consideration of security. There along the Colorado River in the Grand Junction area, all of the access points are immediately adjacent to I-70. This is a very heavily used road with an incredible amount of public access, and we just did not feel comfortable having the technology out in a really obvious place where it could easily be tampered with. We also had a circumstance where it randomly didn't collect 2 samples during the month or so that it was out there deployed. I know Adam, USGS, and Smith Root are working to troubleshoot some of those particular incidences. And I would also highlight that we were somewhat limited by lab processing time. In this circumstance, we would have the auto sampler out there collecting these filters. We would essentially go pick them up once per week. We would then send the filters off to the BOR lab. It would take them 1 to 2 weeks to process those and give us the results. In total, we would get results within 3 weeks in the worst case scenario, which is honestly really efficient. But in this particular circumstance where we only had detections really occurring in a 2-week period, it would be even better if we could get faster results. Next slide. So now with all of this experience, I guess the question would be, are we interested in using this equipment in the future? The short answer to that is yes. To get a little deeper into that, some of my justification is up to this point, detections in the Colorado River have been intermittent at best. We hope that by increasing that sample frequency, we may be able to catch something at the front end of like a spawning event as an example that may allow us to respond more effectively. At this point, the source of these veligers in the Colorado River remains unknown. I am hopeful that if we can overcome some of these security concerns, then we could strategically place these autonomous samplers elsewhere on the Colorado River to give us a little bit more focus in terms of where we are sampling. To kind of address the sample processing time, I've been lucky to work with Stephen Spear a little bit since we started planning for this webinar and talk with him about a little potential collaboration in 2025. I am hopeful that by utilizing both of these technologies in conjunction, if we get something like a positive detection from one of these autonomous samplers, they can get out there with the point of use technology and get much faster results. Really, the hope is that we can get closer to the ultimate source of this thing. And I would kind of close this out by highlighting what others have. I do not view this as a standalone tool in any way. At least in our particular circumstance, any positive eDNA result would trigger additional traditional sampling. We really need to find an organism out there before we would consider any sort of action. That being said, when we are talking about a 75-mile stretch of river, anything that we can do to really limit that area that we are trying to survey on is time and money well spent. So with that, I'll turn it over to the next presenter.
Thanks a lot, Robert. Sorry about that misclick. Thanks for a great example, and thanks for your presentations, Adam. We have a couple more presenters before we move to questions. I see some coming in the chat. Please feel free to keep putting them in there, and maybe our presenters can start taking a look and answering them. Next on the docket are Melinda Barwald and Daphne Gill. They are environmental program managers and geneticists for the California Department of Water Resources. They co-lead DWR's genetic monitoring laboratory that uses environmental DNA for science-driven water management in the San Francisco Bay Delta. And Melinda, and I forget who's going first, but are you good?
Go first.
Yep. Okay, great.
Yeah,
Go for it.
thank you, Kaylin. All right, and you can move to the next slide, please. So we want to start by telling you a little bit about our department, which we'll refer to as DWR. So DWR manages and operates major water infrastructure for California, supplying water to over 25 million people and supporting major international agribusiness. The area we provide water to collectively makes up the world's 8th largest economy. So, continuous operation of our water infrastructure is subject to environmental laws and regulations, including state and federal endangered species acts. Therefore, we are managing the water to benefit both people and the natural ecosystem. And DWR emphasizes the importance of science in guiding our approach to stewardship for California's water resources. It is a core value within our strategic plan. We seek to embrace, support, and collaboratively lead innovations in science and technology as they relate to water resources, including the monitoring and management of invasive species. Next slide. One of the areas in which we are actively driving innovation is the use of eDNA technology for management decisions. A prime example of our commitment to applied eDNA usage is the recent launch of DWR's environmental DNA strategy. The strategy details some of the benefits of eDNA use, including as a complementary expansion to our more traditional ecosystem monitoring activities. Some key management efforts eDNA usage can support, and that includes the continued operations of the State Water Project and several primary applications for eDNA, which includes invasive species monitoring. If interested in learning more, the website for the strategy is at the bottom of this slide. And next slide, please. In terms of invasive species, our current activities include a collaboration with UC Davis to develop point-of-use CRISPR-based eDNA and eRNA assays for early detection of descended mussels, including zebra and quagga, which have not been detected in the San Francisco estuary, as well as nutria, which is a rodent in our system with a large-scale eradication effort underway. So we want to use eDNA and eRNA. eRNA can detect living organisms to identify new locations that they are moving into and the success of local eradication attempts. And next, please. Next slide. Yeah, thank you. I'm sure that everyone has heard of the recent spread of the golden mussels into our system. And we are using eDNA as one tool to determine the geographic spread, with red dots on this map showing locations where either visual detection and or eDNA have been detected for the species. For now, we have not conducted autonomous sampling for any of these eDNA activities, but are interested in doing so and are actively working on collaborations in that regard. Looking into the future, we're also quite interested in MBARI's, Monterey Bay Aquarium Research Institute's work to combine point of use detection with autonomous sampling, and that work is still in development. And so next, Daphne is going to share with you another eDNA study, and this one we did use autonomous sampling for.
Great, thanks, Melinda. Hi, everyone. Now that Melinda has told you about the kind of work that we do here at DWR and our commitment to eDNA, I'd like to tell you a little bit about why we're interested in autonomous samplers. So much like others in other presentations you've seen today, we do a great deal of biological monitoring at DWR, and one of the main goals of that monitoring is to detect rare species such as listed runs of Chinook salmon, delta and longfin smelt, and then also sturgeon. Our sampling often occurs on boats, which means we need crews of several people to do this monitoring, and the monitoring can only occur during the day and at certain times. And so because of these constraints, we may miss detections of rare species, which may be present at night or at other times of the day. And so we see great potential in auto samplers to increase the frequency of our monitoring for rare species, while also decreasing the staff time needed to perform the monitoring. Next slide, please. And so now I'd like to tell you about our first foray into using autonomous samplers for eDNA monitoring with a case study on larval smelt entrainment at water export facilities. As you may know, delta and longfin smelt are two osmerid fish species that are native to the San Francisco estuary in California. And they were once abundant in the estuary, but have declined significantly over the past several decades and are protected under the state and federal Endangered Species Acts. There are many threats to the species, and one source of mortality is entrainment into the South Delta water export pumps that transfer water into the State Water Project's California aqueduct. And if there is a high risk that listed species are negatively impacted by water transport systems, then we have to reduce water exports to protect the species. Next slide, please. So entrainment of juvenile and adult smelt into infrastructure system is regularly monitored at fish protective facilities that are adjacent to the water export pumps. You can see a picture of our fish protective facility here. But entrainment of larval smelts, which are less than 20 millimeters, is not known. And there's a picture here of hundreds and thousands of larval smelt up on the right. So they're very small. And so due to this knowledge gap and to investigate whether smelt take is underestimated, the California Department of Fish and Wildlife mandated that our agency work to produce estimates of larval smelt take near water export facilities as one of our California Endangered Species Act incidental take permit conditions of approval for continued water operations. And much like all of you, we believe that eDNA is a well-suited monitoring complement to traditional net sampling for this mandated work. But we need to test the sensitivity of eDNA to detect this early life stage of a rare fish in our turbid estuary. So we have a lot of challenges in working with eDNA. Next slide, please. So the study that I'll briefly walk you through is a collaborative study with our partners in academia, so at University of California, Davis, and private industry with Kramer Fish Sciences, and then the nonprofit sector, which is the Monterey Bay Aquarium Research Institute, MBARI, which you've heard about already. So before we can use eDNA for monitoring, we conducted pilot experiments to understand how eDNA detection rates relate to, so first, varying smelt larvae abundances, and then second, how well an eDNA signal persists over various distances from an eDNA source. And so what we did was to set up a live car, which in this case is really just another name for fish in a mesh baggie. In this case, a baggie filled with larval smelts. In a channel upstream of the water export facilities. And so you can see here, this is just an aerial view of the channel that is leading up to the water export facilities, where we have a live car here in the orange, and then our sampling distance down here in yellow. And so the idea is that larval smelt shed DNA while they're in the live car, and we then attempt to capture some of the DNA in water samples that we collect further downstream. And so we placed different abundances of larval smelt in the live car and then collected water samples at its 0 to 50 and then 50 to 100 meters downstream. Next slide, please. And so as we investigate these relationships, we also tested 4 different eDNA sampling methods shown here, each with unique benefits for larval smelt eDNA monitoring. However, in the interest of time, and the one that's most relevant for this audience is the autonomous sampler. And so the one we used was the environmental sample processor in the picture in the lower right here, or ESP, which is designed and created by MBARI. And also I should say that we deployed the ESP in a single location in that unidirectional channel that I just mentioned. Next slide, please. So I'm really moving fast through our results here, but I should say that some of our results are still in progress, but the graph on the right shows our detection results from the ESP. So you can see that we have 3 different abundances of delta smelt larvae. So we have 1 larva that was in, again, the live car, 50 larvae, and then 300 larvae. And then our relative concentration of eDNA is on the y-axis. And then again, this is using the MBARI ESP. And what we found is that we were pleased to see that the ESP could detect smelt larvae at an abundance of 300, which is excellent considering the size. So in the lower right-hand corner here, this is a falcon tube, a 50 mil falcon tube with 890 larvae in it. So if you can imagine about a third of that tube being filled with delta smelt larvae, it's again very small. That's what the ESP was able to detect, again in that large channel. And we also only collected 1 filter per time point. That's how the ESP works. You collect 1 filter at a time. And so we were very pleased with the sensitivity of the ESP. However, in the future, we'd like to work on increasing our sensitivity with an auto sampler to detect even lower numbers of larval smelt. Next slide, please. So what worked really well for us with the ESP, well, first of all, was the collaboration with MBARI. If you're lucky enough to get in touch with them and get to work with them, we had just great success working with the team. Kevin Yamahara brought the ESP to us, set it up, and really allowed us and worked with us to program it for our experiments. And it was, anyway, just we feel really lucky to have been to work with this team. Additionally, the sensitivity of the ESP, again, like I told you, we were able to detect 300 delta smelt larvae, which are very tiny in a very turbid estuary. We really liked that we could remotely program the ESP because we inevitably, during some of our experiments, wanted to make some changes. Also, we liked the large capacity for filters. The ESP can hold up to about 120 filters, at least this version of the ESP, I should say. And also it's flexible for filter pore sizes. So we actually tested 2 different pore sizes during this deployment. So a 0.22 micron and a 0.45 micron. Next slide, please. Some considerations for the future and things we'd like to investigate further as we consider our use of autosamplers is the time and skill it takes to set up the autosampler. So we weren't able to set up the ESP ourselves. We had to rely upon the MBARI team, but we'd love to be able to do it ourselves. Also security. So you can see here we have the ESP in a small shed, and then around it we have some fencing. A lot of our work takes place in remote areas that are accessible to the public, and so security and vandalism is a concern for us. Additionally, the size of the sample processor or the auto sampler is something that we would ideally be portable, and then servicing the instrument as infrequently as possible would be great for us, just so we don't have to send teams out there all the time. Additionally, I know in later versions of the ESP, some of the reagents are proprietary, which is difficult for a state agency and our purchasing process. And also, large batteries can be difficult for us to dispose of. So next slide, please. And so in the future, what we're considering for auto samplers is that we may need multiple different auto samplers that are fit for purpose. So thinking about our specific study and what we might need, and then choosing an auto sampler that meets those needs. Next slide, please. And so where we see DWR using auto samplers in the future are a few places. So first, we're really excited because we have a soft mandate, which we believe is the first in the nation that for us to use eDNA for biological monitoring. And so where this would happen is at one of our pumping plants, so Barker Slough pumping plant. And what happens at Barker Slough is regular weed raking. So there's a lot of aquatic veg in front of the pump. So you can see this large rake in the middle here is used to get rid of a lot of that aquatic veg. And then you end up with this pile of aquatic veg, and you may or may not have taken listed species, especially smelt larvae, with it. And so it used to be the job of someone to carefully, painstakingly go through this pile of weeds. And wouldn't it be a huge opportunity for autonomous samplers, and also something that's more sensitive, if we were to deploy an autonomous sampler at Barker Slough pumping plant. Additionally, we have other studies coming up. So we do a lot of habitat actions and species monitoring near our other critical infrastructure. And we just see that autonomous samplers could go there as well. Next slide, please. So as we think about autonomous samplers moving forward, some of the ideal attributes are some that I've already mentioned. So we would love it if they had a remote interface, so we can just control it via our phone. That's the dream. Portability is something that's important to us too. It can be, so ideally our auto sampler could go out on boats or be deployed on shore. We would like multiple filter options, so the ability to use either a Sterivex filter or a Smith Root self-preserving filters and different pore sizes or others entirely. Also, ease of use. We have a lot of field crews, and it would be great if it didn't require extensive training to use the auto sampler. We would love to have abundant filters, again, so we can get at that increased frequency of sampling. And also an adjustable sample volume. We've already mentioned how the estuary is so turbid. We get a lot of clogged filters. So we need to be able to adjust the volume of water that we filter. And then lastly, sample replicates. So this is something that's important to us since we're making it, we need to make decisions based upon some of detections that we might get. It would be ideal to collect more than one filter at a time. So traditionally in our eDNA sampling, we collect our samples in triplicate. And so that's it for me, and I'll pass it back to Melinda.
Okay, so ultimately, what we want to do is routinely use autonomous samplers as a complementary tool to traditional monitoring happening throughout the San Francisco estuary. On the map shown here, there are current monitoring locations, and they're represented as tiny little dots on that map. And apologies that the dots are so hard to see. The reason why they're so tiny is because they are all over the place. And so the scope of our monitoring effort is immense. And it would be fantastic to have autonomous samplers deployed at key monitoring stations throughout the estuary, increasing our species detection capabilities while hopefully decreasing the amount of effort required. Next slide. For that to conceivably happen, we need multiple samplers. So the price point for each needs to make sense, particularly with the threat of vandalism or damage that has been discussed before. And we'd also want the samplers to be scalable, with some sites more frequently sampled than others. So we want it to, be fit for purpose. So for now, we are not using them for widespread monitoring, and instead use them on a based off of specific project needs, such as the examples that Daphne pointed out. Next. Yeah, in our minds, they make the most sense when you want to repeatedly sample the same location over time. And our primary monitoring interests for autonomous samplers and eDNA in general are for the detection of threatened species, invasive species, which we're talking about here, and examining changes in community composition. And we're excited for the tremendous strides being made to enable efficient and effective eDNA monitoring, and I hope to continue in this vein. And next slide. Lastly, we'd like to thank our many collaborators, which span nonprofit, industry, academic, and agency sectors. And we appreciate your time. Thanks.
How about a round of applause for our presenters? We'll move on to questions now, but I just want to say thank you for these fantastic presentations. I'm finding my reaction icon. There we go, clapping. So we do have a couple questions in the chat that came up. And Daphne, I see you already answered one of them, but I just wanted to say it out loud for the group. So how do you deal with clogged filters? Do you switch to a higher filter pore size? Does your target filtration volume vary depending on the location, or do you have a standard minimum volume? Daphne, would you like to answer aloud?
Sure, I can do that. Yeah, it really depends upon the application, but yes, we can, if we know we're dealing with a high turbidity situation, we'll just collect more filters, or we'll use methods that clog less. So we've really been relying on the TONET sampling method for eDNA a lot recently. And it's been very sensitive as a Steravex filter. So we see that as a real win for us. So we've been using a TONET heavily. But our volumes do vary, and but we're even in a turbid environment, and if we collect more filters, we're still able to get kind of a standardized volume.
Thanks, Daphne. And there's another question that came in. It was already answered, but Adam, maybe you could say it aloud and elaborate if you'd like. It was, when there are positive eDNA results but no positive results with any other more traditional methods, how does that inform a response or declaration of invasive mussels present? And this was related to the previous presentation, but yeah, just wanted to give you a chance, Adam, to address.
Yeah, I just wanted to underscore first or just add on to what Daphne said. I think turbidity is one of the current challenges for eDNA autonomous samplers. It's a place that we are working to figure out how best to do it, because just like with manual eDNA sampling, turbidity is an issue with autonomous samplers. Daphne provided how they try to adjust their manual sampling using plankton toad sampling, but it's an active area of development for these autonomous samplers. And it's something that Robert's crew was not directly challenged with, but other partners sampling on the Upper Green River, very high sediment load, were challenged with. And so we did see a lot of fluctuating volumes. And so it's something that I think we're working now with engineer teams to better design an interface that tries to minimize the amount of sediment coming in, or allows us to flush any sediment that comes in to get it out for the next sample. So I see that as being a current area of engineering development, is dealing with turbidity for auto samplers. But with regards to the question you just asked, it's a very similar answer to what was given in the previous set of talks. And I think that Monica and others relayed on this idea that eDNA auto samplers provide similar information as manual sampling, so you can get a detection, but you're still trying to figure out what that detection means relative to the presence of the organism you care about. It's not always a one-to-one relationship. One of the strengths we think eDNA auto samplers have is time series data. So instead of getting a snapshot from a set of samples when you're there once a week or once a month, you can now get a time series of how eDNA concentrations or detections are changing hourly or daily, to help you better understand whether what you're seeing is likely to be indicative of something that's there, or is likely to be indicative of, I guess, what we call exogenous or DNA from a external source. So we think that's one of the great things about eDNA auto samplers. And then to kind of point out a different question that was asked to Robert, in Robert's case, they used this to get more information after they had already had a visual confirmation that there was a quagga or zebra mussel present there in the Colorado River. I think that's really important to underscore. So often we talk about eDNA and eDNA auto samplers from an early detection perspective, but they also provide a really useful means of helping us understand the extent of an invasion or the extent of spread after we get a visual confirmation. So you can use this tool in multiple places in the kind of invasive species management workflow, from is it there to we know it's there, but where else is it, to were we successful with control or eradication. So there's a lot of places where the eDNA and eDNA auto samplers can plug in.
Thanks, Adam. And before we move on to breakout rooms, I want to just check in with my co-facilitator, Mark, if there's anything from the chat you think we should bring up that I didn't.
No, you got him. I was impressed.
Oh, good. Multitasking. And any last questions, you're welcome to, by the way, unmute yourself or raise your hand, but the chat seems to work fine too. Okay, so with that then, I'm going to turn it over to Mark to put us all in breakout rooms. Facilitators and note takers already know their instructions. We have a series of questions. I could put them in the chat right now, but then you would lose them when you went to your breakout room. So I'm just going to leave it to the facilitators to put them in the chat or share your screen when you get to your rooms. Any questions about this? We're good. Mark, are we good to go?
We're good. Yeah, people are shifting around. If you feel like you're in a place that you shouldn't be, I think you should be able to hop back to this main room or certainly send me a note. But here we go.
Thanks, Mark.
Okay.
And I think this is where we pause the recording. Does that sound good to you?
Sure. Are you able to do that?
I see a pause button. I'm going to check it.
Okay, good.
Recording. And so now what we'll do is I hope you had some good discussions, got some good feedback. Thank you, facilitators and note takers. And so what we'll do is we have some time. We have 19 minutes to go around and do some report outs, and we'll have those come from the facilitators. They knew they were going to have to do this. Hopefully you remembered. And share some of the highlights, some of the key themes. And then as we go through, if anyone still on the call, if you have additional thoughts you want to add or questions you'd like to raise, especially questions that we didn't get to earlier during Q&A, please feel free to put them in the chat or raise your hand. You're also completely welcome to unmute and you're also welcome to turn on your video. It seems like we have plenty of bandwidth. So let's start with, I'm going to pick on you, Patrick, because I know that you have a hard stop at the hour. Would you mind going first and kind of reporting out about what your group came up with?
I would not mind. Thank you for that consideration. But I will begin by saying that we ended up spending quite a bit of time on question number 1, didn't get to question 4 or 3, and we just touched on question number 2. So apologies for being a little bit heavy on question 1. We talked a lot about things like numbers of organisms, and one example was given where a single organism was found when sampling for eDNA, when dive samples were done, and how many samples it took to finally get a positive. And there have been some research that, it's pretty obvious that the lower the number of organisms in a system, the more you're going to have to sample to potentially get eDNA. And there's been some modeling work done on that. That might be a need to continue to refine modeling to understand the sampling intensity required. And then things like, what about conditions? We know that an organism that's actively shedding eDNA for whatever reason, because it's in an active feeding period or it's in a reproductive cycle, will require fewer samples to potentially get a positive eDNA. And a question was raised, the eDNA samplers are currently probably limited in how they can be programmed. And it was a question, maybe a suggestion, that if samplers could be designed to be triggered by temperature or some other factor that might be important rather than, take it every hour or every 2 hours. Let's see what else we got. For example, there were some organisms best detected above particular temperature thresholds. Sometimes a disconnect between the people we serve, the citizenry, and the scientists on what we could be doing or should be doing. An example was given of common carp, that the level of concern might not be as high for common carp as for other species or in particular areas because of either lack of concern or lack of understanding of potential negative effects. And on question number 2, I guess that was what I just talked about, understanding gap between managers and the public. And then some discussion on utility of these automated samplers for tracking invasion fronts as opposed to other applications. And at this point, I'll invite anyone from room 7 to speak up if I've missed something important or if I've mischaracterized anything.
Thanks a lot, Patrick. And instead of calling it another group, is there anyone who had a similar kind of conversation or theme come up? We can pass it to you. I'm talking to facilitators. All right, if not, we'll move on to room 2, Adam and Tyler. Adam.
Great. Yeah, thank you. Trying not to be redundant here. I think one of the questions that came up was folks wanted a bit more information on truly how easy training is for the auto sampler. You heard from Robert that, his group thought it was very easy. And then you heard from Daphne for a different platform, the 2nd generation ESP, that it was incredibly hard and required kind of on-site people from Monterey Bay to be on site to do it. So it does run the spectrum. So we got into a bit more of a conversation about the training and about the desire for training for these samplers. Like, one of the specs should be very easy to use. Yes, we talked a bit about that. Spent a little bit of time talking about the range of applications or systems for that our folks in this room could see using it for. There was a handful of folks that really thought that the LAMP tool could really add a lot of value to the program, because right now there is such a bottleneck in turnaround time between when you collect an eDNA sample and when you get those results back. And managers really want information as fast as possible, especially because a lot of these organisms that we're trying to track are moving. And so just because you get a detection on day 1, when you come back 10 days later, it may not be there. So there was a huge need for LAMP. And some of the systems that folks talked about here, invasive species work in the Colorado, doing invasive sampling for the water supply for New York City. And for all these applications, people really cared about early detection and rapid information about early detection. And then there was a question of cost. Like, these tools are great, but they're going to require significant investment if, for example, the participant from New York, they monitor 21 lakes and reservoirs. How many auto samplers is going to be required to do that for the entire program? How many LAMP kits, et cetera? So it represents a substantial investment in equipment, and that is intimidating and likely limiting.
Thanks, Adam. A few of those topics came up in, I ended up in breakout room 1, and cost was brought up, but you all have kind of covered that quite a bit, so I won't go into it. And then also ease of use and training came up as well. I'd like to move on to the next breakout room. Please feel free, if you have additional thoughts on what Adam and Patrick have already said, put them in the chat. Steve.
Yeah, I'll just bring up, I guess, some additional stuff. I mean, one question one question was about the use of the point of use for like airports or like port inspections or shipment inspections, and if that was actually in place. And with particularly bringing up the mossball and zebra mussel example, which, if you aren't familiar, aquarium mossballs or zebra mussels were detected on them a few years ago, and eDNA detection was one thing of interest. And the answer is that there's not currently ongoing LAMP tests at airports developed for the mossballs, but for a variety of reasons that are well beyond eDNA, reflecting like mossball shipments and where they're coming from and all that. It's not actively happening, and I'm not aware of other targets that currently ongoing. However, that it's one of the real potential good uses for these rapid LAMP tests is this sort of this sort of objective. Another thing that was brought up, a sort of different thing with the autonomous samplers. It was a question related to salmon, but really just, is it able to really, is there good examples of it picking up key pulses of activity, such as like a salmon migration? And I noted that actually one of the, I don't know if it's in the wakelet, but we could put it in the wakelet if not. There's actually a published study that with the Mbari sampler with salmon in California showing those differences. So we can add that to the wakelet. Obviously, you could apply that to an invasive species as well. There are also some questions about just eDNA in general, such as like residence time of eDNA in different types of environments. I think another kind of comment just on, thinking about use cases, talked about like putting out, it would be really helpful to have auto samplers across like an entire, state area to do all sorts of invasive species surveillance. But currently, there's not really the infrastructure, at least in a lot of places, to how you would analyze all of those. Certainly that relates to some, some of the things with like the molecular lab network, but more broadly, that can be a holdback. Although in the future, if these become truly autonomous with the analysis happening along with the collection, that would, address a lot of those issues. Yeah, and then I guess the last thing, and this was already echoed, is that time is, for point of use, time is really the key thing. The fact that you can get that turnaround result is really would drive a lot of interest in that. I don't know. I think I hit on most things that we discussed. Yeah.
Yeah, thanks, Steve. And we have a little bit more time, so we're gonna try to finish. There's 3 more breakout rooms. Devin, then Nick, then James. Maybe 2 minutes each.
Yes, I'll be very brief. I think one of the biggest things that our room talked about was cost. So it seems like every state or department, whatever, is sort of limited in their manpower and budgets as usual. And so one of the things that came up is something that would be really useful is just being able to leave a sampler out for a long period of time. So not having to worry about preservation and sample quality, but then also having a lot of samples to leave things out for longer would be really, really helpful for some of those groups that don't have as much time, money, manpower, et cetera. And then the other big concern was just thinking about how we can have these samplers that can go from a very turbid lake or location to one that's not so turbid, and making sure that it's useful for these different types of uses.
I think we've had a pretty good overlap, especially in question 1, so I'll probably just skip over that. Some interesting things that were discussed in question 2. In Colorado, auto sampling has allowed them to kind of expand from just reservoir sampling to include river monitoring. There was in the group some pretty common interest in applying point of use assays to terrestrial species or specifically invasive reptiles. The existing tools that they have aren't particularly rapid, so it would be nice to have some more rapid type approaches. And there was some discussed interest in using auto sampling, especially in small lake systems where they're specifically monitoring for a single or just a few invasive species. Question 3, there was quite a bit of overlap as well, but having clear and standardized sampling designs or protocols available for these approaches would be useful, especially how many sites and over what time frames are needed for best use. Also, more cost-benefit work, assessing trade-offs between the available approaches or more traditional methods. And again, some more interest in applying these things to terrestrial systems.
Gosh, I remember who was in your breakout room. All right, Olivia is going to report out for room 6. Thanks, Olivia.
Hey, yeah, I think our group pretty much echoes a lot of what it's already been said. I'll summarize briefly. Anyone from our group, feel free to like correct me or add, I think, or throw something in the chat if you need to. But yeah, I mean, some similar concerns came up around the cost, especially the auto samplers, and making sure and wanting to understand better, like what, how much you kind of get from that, you know, kind of price of unit and that kind of thing. Overall, yeah, there was some, you know, concern about the training and the time and everything required for that. And a lot of, you know, people could see a lot of different applications for these kinds of tools. There was some talk around like needing it for like port situations and needing that kind of real on the ground, quick, immediate data. So there's a lot of discussion around those challenges too, of like, we need what we, you know, as much data as we can get right now, and also need to be able to trust that data. And the concerns around like false positives and wanting to understand better about the different methods and making sure we understand when and where false positives come from and like what to do with those and all that good stuff. So a lot of those kinds of conversations. And we also only got pretty much to the first two questions. Yeah. Trying to see if there's anything else maybe to summarize for us. There's, yeah, I'll leave it at that, and people can throw in a little more if there was something big I, you know, missed from the, yeah.
Yeah, thanks. That's interesting. I'm hearing kind of like the different pros and cons of both of these tools, which is kind of fun that we got to put them into the same webinar. One is pretty quick, pointed, and one can be dispersed and really save some manpower. So I see Adam put in the chat, virtual training for the Smithford eDNA auto sampler, which is about an hour, includes a lot of chit chat discussion.
Just meaning like a lot of like back and forth kind of questions versus like step 1, step 2, step 3. It's just, you know, it's everything is done, including kind of, you know, just curious conversation, small talk in about an hour.
Gotcha.
So we've done that now successfully with a handful of partners at about an hour, and it's comparable on site. It's about an hour.
On site. Oh, okay. Yeah, interesting. There is a fun, my group did get to the 4th question, which was filling in the blank. I want to use an eDNA auto sampler for, I want to use point of use for. And I liked this response. I personally like a system that was both point of use and autonomous, that remotely transmits results back to us. Not a reality for now, but that would be a game changer for us. So I thought that was nice. It married the two nicely together. That would be the dream. So next steps, we have 2 minutes. We have a number of different resources we've referenced throughout the webinar. I put in the Wakelet link again. That will have the 3 previous recordings and eventually this recording. And we have, because this was a meeting where you all registered for it, we have your emails. So we will send a follow-up email to everyone who registered, not just attended, with a summary of the notes that we have from the discussions, as well as the recording, which will be posted on that Wakelet, but also on the AFWA YouTube channel. So I just want to thank you very much for your participation and sticking with us all the way to the end. We still have 52 people on, so that's pretty good. Thank you very, very much to all of the facilitators and note takers and presenters. Definitely couldn't do this without you. And I think that's a wrap. I gave you 1 minute back.
Thanks all.
Thanks, everyone.
Thank you.
Thank you.
Bye-bye. Thank you.
Thanks.
Hi, Karen.